thank you for joining me on UCSD Guestbook. Tonight it is my pleasure to welcome UCSD Professor Robert Galambos, an eminent neuroscientist and one of the founders of the neuroscience community here in La Jolla. Welcome, Bob.
Thank you.
Bob is an extraordinary scientist whose research laid the groundwork for much of our understanding of the auditory nervous system, that part of the nervous system that deals with hearing. Key features of our basic understanding of the neurobiology of audition, of hearing, came from his pioneering work, and he has also developed several important clinical tests to detect hearing deficits. Bob, as a graduate student at Harvard in the 1940s, you became very interested in whether bats could hear the very high-pitched sounds that they had just been discovered to make, and whether perhaps this hearing process was part of their uncanny ability to avoid obstacles. Tell us how you came to these experiments and how they unfolded.
The year is 1939, and Don Griffin and I are graduate students in the biology department at Harvard. He had just discovered that bats make high-frequency sounds that we can't hear as they fly around, and he wondered whether they could hear them. At that time, I was working with Hal Davis in the medical school on the other side of the river on the hearing of various animals, guinea pigs, for example, using a test which is called the Cochlea response test. This is a measure of their hearing. So I asked my boss if we could put a bat in the place of the guinea pig, and he said fine, which I did. And within a week, it was clear that these animals have an extraordinary ability to hear sound frequencies above the level that we were able to measure with our machines.
Mm-hmm. Did the bats object in any way to being immobilized, or were you actually capturing them as they were flying about?
Well, Don was the expert in capturing them, and he had a small colony of them. So I took a couple of them in a little box by a subway across to the medical school, anesthetized them, of course.
Ah, there we are.
Yes. And then proceeded with the measurements. Well, Don and I then took the bats into a soundproof room in which there were wires hanging from the ceiling and measured their ability to avoid these obstacles as they flew. And, of course, we found they were helpless if their ears were plugged or if their mouths were tied shut so that they couldn't, they remained mute. And that's the story. That story became my PhD thesis in 1941.
Well, that was only part of your PhD work, Bob, if I remember correctly, because I think you went on with Hal Davis to work further on auditory neurons and discovered what I think remains to this day one of the fundamental properties of these auditory neurons in the cochlea, that they have a best frequency, a particular tone to which they respond best. Tell us a bit about that work.
Well, that's interesting. At that time in this medical school physiology department, Birdsie Renshaw had just used microelectrodes to get responses from the hippocampus. And I was there watching what was going on and was interested in using the microelectrodes in some way. And I remember very clearly the day it happened. I was back in my office in the biology department, and it struck me that this would be a way to resolve a long-lasting problem of auditory physiology. There were two main schools of thought about how the ear worked. One was the Helmholtz idea, which was that the cochlea is a structure which is tuned like a piano to various strings. So this was one idea. Whenever the sound came in, it would activate only some of the auditory neurons. The other line of thinking was that all of the neurons responded at the frequency of the stimulus. So here was a classical setup. It occurred to me, all I have to do is to put the microelectrode into the auditory nerve, and the nerve will either respond to one tone or a few tones, or it will respond to all of them. And all nerves will respond to all tunes. That was the thought. Fairly simple-minded, don't you think?
Well, but simplicity is the key to success so often in science.
So I went to my boss, Hal Davis, and told him this. He said, you know, we've been trying to do that experiment around here for years.
Do you have the temerity to think that you can solve this problem?
Something like that. And I promised him then, it was, as I remembered, in the fall, I promised him that I would have the answer by Christmas.
Whoa.
I'm happy to say that I did. I laid the first auditory tuning curve on his desk on Christmas Eve. The tuning curve, as you have just said, is the relationship between the frequency of a stimulus and the response of the unit in some measure or other. And this is the tuning curve. Well, that's where the tuning curve was invented. And you'd think now that that would be the resolution of the classical problem, you know, the classical dichotomy. Well, it turns out that while each unit likes to respond best to some frequencies, if the frequency is low enough, the unit will respond at the frequency of the stimulus. So both ideas were correct. And from that point on, you never heard any more about this fundamental dichotomy.
This points up a wonderful general feature of science, I think, Bob, that so often when one has two opposing camps that hold radically distinctive views, the answer lies somewhere in the middle or by combination of the two views.
That's right.
And your work illustrates that beautifully.
Yes, that's the way it works.
One might argue that success in science is a result of being in the right place at the right time. Many people hold this view. On the other hand, there's something to be said for Pasteur's dictum that in the field of science, chance favors the prepared mind. And I wanted to ask, in your own case, in your own history, was it a matter of being in the right place at the right time, or were you perhaps very well prepared to be receptive to and to be creative about solving these important problems?
Well, I'm sure you realize the answer is both. Let's take this bat problem. That was 150 years old, and some big minds had attacked that problem, and they have not been successful in resolving it. And when you think about it, that problem can be solved only if two conditions are met. The first one is that somebody has to invent machines that will produce sounds that we can't hear and will measure them. And Don had worked with a professor who had accomplished this. And the problem also requires that you have a good physiological test of the hearing for testing the hearing of an animal. And that had just been resolved by my professor, Hal Davis, in the medical school. So here we are, two eager graduate students who have, for the first time in their hands, the two requirements for solving the problem. And given all that help, well, of course, our professors also were happy, were competent to address our questions, obviously, by their various inventions, and were happy to have us go to work on these. So Don had a lot of skills and information that I didn't have, and vice versa. And the two of us together could take advantage of this wonderful opportunity that a great university gives its graduate students.
Yes. Well, those Galambos and Griffin papers are classics that are read by students today. So you certainly should be very proud of that incisive work at such an early stage in your career. Many scientists seek during their careers to make a contribution that gives something back to society, something that will be beneficial to the population at large. And in biology, often this is some kind of a clinical contribution, something that has an application for healthcare in the populace. You've been very successful in such contributions. Could you tell us a little bit about how you got started with pioneering clinical tests for hearing deficits?
My interest in hearing tests dates back to the time I worked for the United States Army as a civilian in Washington, D.C. at the Walter Reed Army Institute for Research. I was the boss of the neurophysiology unit there. And the young men who came to work in the place were captains in the medical corps who had careers in research before they were inducted to spend their 2 years. Well, we heard about such people and were able to select the ones that we would like to have come and work for us for a kind of a postdoc for 2 years. One of these, by the way, is Dave Huebel.
Dave Huebel,
Dave
yes.
Huebel, and the first 6 papers that won him and the Nobel Prize were done in this Walter Reed lab. Well, the first thing I did when these young men came was to give them what I call the 1/3, 1/3, 1/3 rule. I pointed out that they were working for the United States Army, and so it would be all research. But something like 1/3 of it they might think of as payback to the Army. And the Army had numbers of problems that needed solution, and they should think how their research might contribute to that. Another third, I told them, was to find out who else was working in this place and might have skills that they didn't. And the two of them together could perhaps come up with some experiments that couldn't be done by either of them alone. Shades of working with Griffin, of course, yes. Same idea. And finally, the third should be entirely their own to do whatever they wish to do. Well, taking my own advice, I looked around for a hearing problem in the Army, and there is one. There are some soldiers who fake being deaf.
Goodness.
With the idea that when they get out, they will receive a disability pension for the rest of their lives. And these should be distinguished from the soldiers who are really deaf or have hearing loss. So I worked out a couple of tests that did distinguish the malingers from the non-malingers. One has to do with the number of times your eyes blink when loud sounds are put into the ears. They don't blink until the sound is a certain level, and then they continue blinking above that. And this is a very good test, and it will distinguish.
Caught the malingerers.
That's right, it will catch them.
Very nice.
The other one was an EEG test, but these tests were kind of complicated to perform in a busy clinical situation.
You went on, Bob, to develop a test that is non-invasive, that can be administered even to, I think, individuals who are asleep. I'm thinking of the auditory brainstem response here. Could you describe that response?
Yes, well, I'm coming to it.
Okay, very good, very good.
Because these experiments were done in the 50s, middle 50s or so. And so we jumped to about 1970, 72, somewhere in there, where a preprint of a paper by Jewett and Williston, a couple of San Francisco people, came across my desk. Well, now, Jewett had worked with me as a postdoc at Yale. And while there, he discovered that, incidentally, we had some of the first computers being applied in the 70s at Yale. in labs, and he was recording from the skull of a cat and putting clicks into the cat's ear. Well, now, as you can imagine, the auditory system moves from the ear ball up to the midbrain, up to the cortex. And he discovered that he could see this entire train of events with the electrodes on the skull of the cat, a anesthetized cat, of course. And when he left for his new job in San Francisco, he carried with him the idea that this ought to be something that would work on people. And this was the first paper in which he presented his first results on the brainstem response. Well, about that time, I was here at UCSD and doing some joint work with people down in Children's Hospital, where one of the problems is, obviously, do newborn babies, what is the hearing of newborn babies? Well, one of the standard tests was to drop a bedpan on the floor.
Simple, but perhaps...
Simple. And another one is to slam the door behind the kid's back.
Yeah.
Well, we undertook this, we grasped the idea that maybe this Jewett and Williston brainstem response was the test that would work.
You developed this as an ongoing procedure, if I remember correctly.
We investigated. We went into the newborn nursery and tested them right in the newborn nursery. And the answer there was that newborn babies don't have any hearing loss. Normal babies have normal hearing. But then we went into the intensive care nursery, and there's a lot of auditory pathology there, which we worked out. And after several years, the administration at Children's Hospital accepted our results and set into the operation the requirement that before the infant is released graduates from the intensive care nursery, he used to have one of these brainstem response tests. And that's where all the pathology is. Something like 15% of the babies who leave have hearing loss.
Gosh, yeah.
And then we followed them up, of course, in the Speech, Hearing, and Neurosensory Center down at Children's Hospital with Don Krebs and Bob Sandlin. We followed them up then in the hearing clinic. That program is still underway 25 years later down at Children's Hospital. As I understand it, is still identifying something like 2% of the babies who have permanent hearing losses need hearing aids.
Hearing aid, I was going to ask about hearing aids, and this is a nice lead to that. These are remarkable devices that can do an enormous amount to ameliorate a variety of hearing deficits, but some of the difficulties, particularly those that are more central in the nervous system, make one wonder whether it will ever be possible to have a fully artificial ear that connects then and in some way transduces sounds meaningfully to the auditory cortex in the brain. Do you think that will become a reality at some point?
Well, you know, the cochlear implant story it comes close to what you have in mind. Electrodes are put into the cochlea at different places, and they're connected to the microphone and a little stimulator, which decides which frequency should be stimulated within the cochlea itself. So that that device is commonly used now in adults as well as in children. This is the sort of thing you have in mind.
Yes. Well, that's a very important...
It works.
Advance. In recent years, you've moved your research away from audition and hearing to the visual nervous system, and you have made a series of very interesting results that I think have the promise of reorienting our understanding of the way information moves from the retina of the eye through various centers on its way up to the cortex. How did you become interested in this problem? It's so different from your earlier work.
Well, I closed the door on my last lab here in 1982. Then I struck up a friendship with a Hungarian in Budapest, Gábor Juhász, who has a laboratory. He's actually a biochemist, but he's also interested in electrophysiological matters. We talked about some joint experiments. I have to skip here several years to come down to the point that you wish me to make. Visual system. Now, I've spent all my life as an auditory physiologist, and I knew enough to give lectures to medical students about the visual system, but I really didn't know anything about the visual system.
All we have to do is stay one step ahead of the students.
Yeah. Let's jump to 92, because that's when Juhász and I thought we knew enough about the visual system to implant some rats with electrodes in the eyeball, at the optic chiasm, which is where the optic nerves cross, and in the visual cortex of the rat. Well, now, implanting animals of this sort was an old story by then, but the new thing that we did was to glue an LED onto the skull.
Light-emitting diode, generate
Light-emitting diode, so that every time we push a button, there's a flash of light that illuminates the skull of the rat. And we could show that this illumination, which of course we could vary in intensity and in duration and in number that we give, all these dimensions now were possible for us to give to the rat. And that was the key to a preparation that is stable and very useful indeed. Any time of the day or night you so impose, you push the button and your preparation is, the rat carries his own stimulators. A very useful preparation. So we spent several years working on this preparation, coming to understand the properties of the visual system, many of which were not the properties of the visual system that
had been previously studied
in the textbooks. Textbooks.
But Bob, you focused on the way in which the eye automatically, whether we like it or not, jumps from one site to another, essentially chunking information about the visual world and then relaying it up to the brain. Is that the sense of it?
Exactly. Now, you know this very well from the fact that you know what happens when you read. You look here, then you look there, and then you look there, and you move your eyes 3 times a second from one spot to another when you read. And both eyes do the same thing, and in 1 minute you have made 400 of those fixations between the saccades. Now, it follows, without any discussion, further discussion, that each one of those fixations creates a rod and cone pattern of bleached white and black, representing the spaces in the letters.
These are the cells in the retina that are exposed to the light.
And then they convert this into optic nerve volley and send it upstairs.
Electrical signals going upstairs.
That's right. So if you know this to be true, and everybody knows this to be true and knows it for 100 years to be true, then the way the system operates at the beginning is to send, this is 3 hertz, send 3 complete optic nerve volleys to the brain every second. Now,
this is amazing, Bob. You know, the key to success in science, I think it could be argued, is looking at what everyone is looking at, but seeing what no one has seen. And I think your recent work here is a wonderful illustration of that.
Well, I think that's right, because if you pursue this idea, you come to the conclusion that a lot of the experts are going to have to change their model of the visual system. It is not a system in which there's a steady stream going upstairs. It's a
series of snapshots, effectively, being chunked and sent up to the brain.
Now, the question is this, so this is what happens in reading. Is it also what happens under ordinary circumstances. So someone I found a paper, someone in England had instrumented an automobile driver, and sure enough, as he drives through the streets of London, his eyes are moving on the average 2.5 times a second.
It must happen everywhere. Bob, our time is running short, unfortunately. I have the feeling we could be talking for hours together here, but there's one question that I very much want to ask you to kind of focus on your forward view of neuroscience. If you had the ability to ask a single question, just one question, of some perhaps almighty power that could grant you the answer to this question, what question would you ask?
Of course. How does the brain work?
Would you focus particularly on timing of information and how it's relayed into the brain? This seems to be perhaps a common feature both of your work on the auditory system and on now more recently the visual system.
No, it's a different problem from that. There's a bat that's 1.5 inches in size, and he does all of the things that a bat does. He echolocates, he knows how to reproduce, he knows how to take care of himself, how to find food, and all of the things that every other organism knows. Propagate, save yourself and propagate the species. And then think about the ant. The ant also has a brain. You
want too much, Bob. You want too much.
But that's the question.
Those are the questions. Yeah.
That is the question.
Yes.
How do the genes put together a system that's universal in its placement in organisms and that fits the individual exactly for the environment in which he exists? Yeah. That's the question. How does the brain work? Because the brain is what does it.
Certainly is. Thank you very much, Bob, for joining me this evening. Thank you for joining us on UCSD Guestbook.